Imagine this hidden microscopic enemy. It's literally everywhere in the soil, the water, you name it. Yet it only really strikes under certain conditions. And that's what's so fascinating. This bacterium, it's found globally, super resilient, but its ability to cause disease is, well, it's quite nuanced. Today, we're doing a deep dive into listeriosis. That's the bacterial infection caused by Listeria monocytogenes. And we're focusing specifically on its impact on two pretty interesting groups of animals. Camelidae, so camels, llamas, alpacas, and Cervidae. That's our deer, elk, and moose. Our mission today, to really unpack this tough little pathogen, understand how it infects its hosts, how disease actually looks in these unique animals, and maybe what its presence tells us about, wider animal and even human health. We've got a great veterinary review article, plus some insights from other recent research to kind of guide us.
Yeah, it's a really fantastic example, actually. It shows how things like the environment, the way we manage animals, even the health of the whole ecosystem, how they can all intersect through just one single type of bacteria. You'll see how this one, common bacterium, can paint entirely different pictures, epidemiologically speaking, depending on which animal we're talking about and what kind of environment it's in.
Okay, so let's zoom in a bit.
hmm
What exactly is this bacterium, Listeria monocytogenes? It sounds pretty formidable.
It absolutely is. I mean, technically, it's a small, motile, gram-positive, non-spore-forming cocobacillus. But what's really striking is its incredible environmental resilience. It's what we call a ubiquitous saprophyte. Basically, it lives on decaying stuff, and you find it everywhere: soil, water, manure, rotting plants, globally.
So it's basically unavoidable out there. What makes it so incredibly tough?
Well, its basic physiology is just remarkably hardy. It's psychrotolerant. That means it can grow across a huge temperature range from almost freezing, like 0.5 degrees Celsius, up to about 45 degrees. And that ability to grow in the cold, that's key. It means it can multiply even in refrigerated food. We call that cold enrichment. That's a big reason why it's such a headache for food safety. Plus, it handles high salt, a wide pH range, and, importantly, it can form biofilms. These are like sticky shields that protect it from disinfectants, makes it really hard to get rid of on surfaces.
Okay, that environmental toughness is impressive. But here's what really gets me. How does something built for surviving in dirt then cause such specific bad disease inside an animal? It's not just floating around in there.
Right. That's the transition. It's an invasive intracellular pathogen. It doesn't just hang out between cells. It uses these specialized surface proteins, internalins, like NLA and NLB, to bind to host cells. It basically tricks the cell into engulfing it, taking it inside into a little bubble called a phagosome.
So it gets itself captured, essentially. But then,
Exactly. But it doesn't stay trapped. Once it's inside that phagosome, it uses toxins like Listeriosis O or LLO and some phospholipid pests to blast its way out.
so it breaks out of that initial prison.
Precisely. Now it's free in the main part of the cell, the cytoplasm. And here's the really clever bit. It hijacks the cell's own machinery, the actin network, to build itself a kind of comet tail. This tail literally pushes it through the cytoplasm and even propels it right into neighboring cells. It spreads from cell to cell without ever having to go outside where the immune system could easily find it. And this unique cell-to-cell spread, that's fundamental to how it gets across critical barriers: the gut wall, the blood-brain barrier, the placenta.
Wow, so it breaks out, uses the cell's own engine to move, and avoids the cops, so to speak.
Yeah.
Incredible. And is it always the same type of Listeria doing this, or are there variations?
Good question. That raises an important point. Listeria monocytogenes isn't monolithic. It's genetically diverse. We see specific strains or groups of related strains we call clonal complexes, often associated with particular forms of the disease. For example, strains from clonal complex 1, CC1, they seem to be significantly linked with the neurological form, suggests they might have a preference, a sort of neurotropism. Others might be more likely to cause abortions.
Okay, so we have this resilient, clever, diverse bacterium. Given that, what actually triggers an outbreak in animals like camels or deer? It can't just be random chance if it's everywhere.
You're absolutely right. It's usually not random, especially in farmed animals, including managed camelids and cervids. Clinical listeriosis is very strongly linked to specific farming practices, so much so that it's often called silage sickness.
Silage sickness. Okay, tell us more about that. What's the connection?
Well, outbreaks are very frequently traced back to animals eating poorly fermented silage. Particularly silage where the pH hasn't dropped low enough Maybe it stayed above 5.0 or 5.5. See, the whole point of making silage is that fermentation should rapidly lower the pH. That creates an acidic environment that preserves the feed and stops spoilage bacteria, including Listeria. But if that process fails, maybe the silo wasn't packed tightly enough, air got in, the forage was too wet or too dry, the higher pH lets Listeria multiply like crazy. Then, maybe 10 to 21 days after the animals start eating that bad batch, you see the disease pop up.
So in these managed herds, it really sounds like it's often a disease of, well, management failure.
Precisely. The pathogen is, pretty much always around in the environment. But the actual clinical disease, that tends to emerge when something goes wrong with feed storage or maybe hygiene, allowing the bacteria to build up to a dose that just overwhelms the animal's defenses.
And what's interesting here is, thinking about it, it's probably not just the feed itself, right? The whole environment must play a role.
Right, absolutely. Healthy animals, even ruminants, can be asymptomatic carriers. They can carry L-monocytogenes in their intestines and shed it in their feces for weeks without looking sick at all. This constantly recontaminates the soil, water, feed bunks. It sets up this fecal-oral enrichment cycle that keeps Listeria established on the farm. We also see seasonality peaks in winter and early spring, which ties right back to when animals are most likely to be fed stored forages like silage. And yeah, general stressors matter too. Sudden cold snaps, overcrowding, the stress of late pregnancy. All these things can make animals more susceptible if they get exposed.
Okay, so once an animal is exposed and overwhelmed, how does listeriosis actually look? You mentioned different forms earlier.
Indeed, the clinical presentation is really a direct result of how the pathogen gets in and where it goes. We generally see three main forms: neurological, septicemic, and reproductive. And it's interesting, usually one form tends to dominate during a particular outbreak.
Let's start with maybe the most dramatic one, the neurological form, sometimes called circling disease, right?
Yes, that's the one most often recognized in adult ruminants. And its pathogenesis, how it develops, is quite specific. The thinking is that the infection starts from tiny cuts or scrapes in the mouth or maybe the nose, often caused by eating rough or coarse feed.
And from a little cut in the mouth, it travels all the way to the brain. How does that happen?
Yes, it takes a very specific route. The bacteria seem to get into the nerve endings right there in the mouth or face, especially the trigeminal nerve, that's cranial nerve 5. And then they travel backwards inside the nerve fiber itself, what we call retrograde axonal transport, directly to the brainstem. That's the pons and medulla oblongata area. This results in this localized, often just one-sided infection in the brainstem.
That's incredible, traveling along the nerves. So what would you actually see in an animal suffering from this?
The classic signs are pretty distinctive. Depression, disorientation, maybe circling constantly in one direction. Head pressing is common, pushing their head against a wall or fence. And those unilateral cranial nerve problems. A drooping ear, maybe a droopy eyelid, paralyzed lip on just one side. They might also be unsteady, ataxic, have trouble swallowing, maybe see some flickering eye movements. It can progress to the animal going down, being unable to rise, even seizures. And importantly, the incubation period for this form is much longer, typically 3 to 7 weeks.
Wow, quite a specific and devastating picture. What about the other forms?
Well, the septicemic form is different. It usually starts when an animal swallows a really high dose of Listeria. It invades through the intestinal lining and gets into the bloodstream. This bacteremia, bacteria in the blood, can lead to a very rapid, often fatal systemic infection, septicemia. This is especially common in very young animals, neonates, or those with weakened immune systems. Clinically, you'd see things like fever, just general lethargy, no appetite, and if you looked post-mortem, you'd find tiny spots of dead tissue, necrosis, scattered in organs like the liver and spleen. The incubation here is short, just a few days.
And for pregnant animals, you mentioned a reproductive form.
Yes. In pregnant animals, if the bacteria get into the bloodstream, they show a strong preference, a tropism, for the pregnant uterus. It colonizes the placenta, causes inflammation there, placentitis, which ultimately leads to the death of the fetus, typically abortion late in pregnancy, usually the last trimester. Often the mother animal, the dam, shows very few signs herself. Maybe she's a little off her feed, but often the abortion is the first major sign something's wrong.
Okay, so we have these distinct patterns of disease. Bringing it back to our specific focus animals. What does all this mean for camels, llamas, alpacas, and deer? How does listeriosis show up in them?
Well, the interesting thing is that the basic disease process seems to be very highly conserved across these different ruminant and pseudoruminant species. The clinical syndromes we just discussed, neurological, septicemic, reproductive, they appear remarkably consistent.
Okay, let's start with the Camelidae then. Camels, llamas, alpacas.
Right. So clinical listeriosis has definitely been documented in both the Old World camelids, like dromedaries, and the New World ones, llamas and alpacas. Now, reports of actual clinical disease are maybe a bit sporadic compared to sheep or cattle, but serological studies looking for antibodies in the blood suggest exposure is actually pretty common. One study in Iraqi camels found nearly 20% had antibodies.
So they're definitely encountering it, even if clinical disease isn't reported constantly. When they do get sick, what does it look like? Similar signs.
Very similar. The neurological form is the most well documented. There was a case report in dromedary camels describing these sort of Parkinson's-like head tremors, paralysis of the lower lip, incoordination, and it progressed rapidly. In llamas and alpacas, you see the classic stuff. Circling, head pressing, fever, ataxia, those one-sided cranial nerve deficits, drooping ear, lip, eyelid. And yes, the septicemic form and the reproductive form, including abortions, have also been reported in camelids. It really mirrors what we see in other ruminants quite closely.
Okay, and switching gears to the Cervidae family are deer, elk, moose. How does it affect them?
Listeriosis is known to affect a pretty wide range of cervids, both wild and farmed. In farm deer operations or maybe park settings where they get supplementary feed, outbreaks often look very similar to what we see in cattle or sheep. They're often linked to feeding practices like silage or environmental stressors. There was one notable outbreak in fallow deer linked to a combination of stress from a poor natural food year, maybe higher stocking density because of that, and then a sudden cold snap. Again, those predisposing factors coming into play.
But connecting this to the bigger picture, what about wild cervids? They aren't lining up at a silage bunk? Does it play out differently there?
Ah, yes. This is where it gets really interesting and potentially quite different. Wild cervids are clearly important environmental reservoirs. Studies have found listeria in the tonsils of a surprisingly high percentage of hunt deer, for example, even when their feces were negative, so they can definitely carry it. But clinical disease, especially the fatal septicemic form in young wild animals like fawns, seems to be strongly associated with concurrent infections, particularly immunosuppressive viral diseases.
So for wild deer, Listeria might not be the main aggressor. It's more like an opportunist jumping on an already weakened animal.
Exactly. That seems to be the pattern more often in free-ranging wildlife. Listeria acts as a secondary invader, capitalizing on a host whose immune system is already compromised by something else. So this kind of positions listeriosis in wild deer less as a primary disease driver and more as a potential sentinel. Its presence, especially causing death, might signal that there are underlying immunosuppressive diseases circulating, or maybe broader health stressors affecting that population. It's a clue to look deeper.
That's a really important distinction. Okay, so whether it's a farmed llama or a wild deer, if listeriosis is suspected, how do vets actually pin down the diagnosis? And crucially, can anything be done?
Well, a strong presumptive diagnosis can often be made just based on the history, especially that link to poor silage in farmed animals combined with a good neurological exam showing those characteristic asymmetric brainstem signs. But for a definitive diagnosis, you really need laboratory confirmation.
And what are they looking for in the lab? What confirms it?
Postmortem examination is really key, especially for the neurological form. You need brain tissue. The pathognomonic lesion, the sort of telltale sign, is finding these specific multifocal microabscesses and mononuclear perivascular cuffing, basically inflammation around blood vessels, primarily in the brainstem. Then, definitive confirmation comes from actually isolating L monocytogenes through bacterial culture from the affected tissues, like the brainstem or maybe fetal tissues in abortion cases. Sometimes, because the bacteria can be tricky to grow, labs use that cold enrichment technique we mentioned, refrigerating the sample broth for weeks to help the listeria outcompete other bacteria before plating it out. Molecular methods like PCR are also increasingly used for faster detection directly from tissues.
And to treatment, is there hope for recovery?
Treatment needs to be prompt and aggressive. High doses of parenteral antibiotics, injected antibiotics, usually penicillin or oxytetracycline, are the mainstay. Supportive care, like fluids and anti-inflammatories, is also critical. However, the prognosis really hinges on how severe the signs are when you start treatment. If you catch it very early, before the animal is down, recovery rates can be, maybe up to 50% in some species. But once an animal is recumbent with advanced neurological signs, the prognosis is generally poor to grave. And of course, for free-ranging wildlife, treatment isn't really a feasible option.
So it really sounds like prevention is where the focus needs to be, especially for managed animals.
Absolutely. Prevention is by far the most effective strategy, particularly in managed herds, and the single most critical thing. Avoid feeding spoiled or poor quality silage, period. Only silage that is properly fermented down to a pH below 5.0, ideally lower, should be fed. Any silage that looks moldy, smells off, or comes from those outer exposed layers of a bunker or pile, that stuff should just be discarded. Don't feed it. Regular cleaning of feed bunks to remove old feed and avoiding feeding directly on the ground where contamination is likely, those are also really important basic hygiene measures.
It's fascinating how these basic management principles remain so crucial. So summing that part up, what does Listeria tell us about the bigger picture of animal health?
I think it tells us we need to be constantly vigilant about the interplay between the environment, the feed, and the animal's own susceptibility.
Yeah.
Listeria is this master survivor, this opportunist. Seeing it cause disease often points to a breakdown somewhere else in feed management and environmental hygiene, or maybe underlying health issues in the animals themselves, especially in wildlife.
And there's another layer here, is that this isn't just an animal health issue. There's a human connection.
Oh, absolutely. You're spot on. Listeria monocytogenes is a major foodborne zoonosis, meaning it passes from animals to people. And it's considered one of the most serious foodborne pathogens because, while less common than something like Salmonella, it has very high rates of hospitalization and mortality, especially in vulnerable groups. We're talking about pregnant women, newborns, the elderly, and anyone with a weakened immune system.
How do people typically get infected? Is it mostly handling animals or food?
The main route by far is consuming contaminated food. And the list is pretty broad. Things like unpasteurized milk or soft cheeses made from it, ready-to-eat deli meats, smoked seafood, even sometimes contaminated raw vegetables. But there is also an occupational risk. Farmers, veterinarians, slaughterhouse workers, anyone having direct contact with infected animals, particularly aborted fetuses or placentas, which can be teeming with bacteria, they are at higher risk of direct infection.
So our circus animals today, the camelids and cervids, they definitely play a role in this cycle.
They certainly can, yes. As we discussed, asymptomatic carriers shed the bacteria in their feces. This contaminates pastures, water sources, potentially farm environments. That contamination can then find its way onto raw agricultural products like vegetables. And, of course, meat and milk from these animals, if not properly handled pasteurized, cooked thoroughly, can be a direct source of infection for people. Finding pathogenic Listeria strains in wild deer really underscores their role as environmental reservoirs, potentially contributing to contamination cycles that could impact public health indirectly.
This really drives home the interconnectedness of it all, doesn't it? Animal health, human health, environmental health.
It absolutely does. It's a textbook example of why we need a One Health approach.
Yeah.
We have to monitor pathogens like Listeria at that interface where wildlife, domestic animals, and humans interact. Understanding those transmission dynamics is crucial for protecting everyone.
Wow. What a deep dive today. We've really peeled back the layers on Listeria monocytogenes. It's this incredibly tough, widespread bacterium posing quite a unique threat. We've seen how it causes silage sickness in farm camels and deer, where it's often tied to management, but also how it might act as a kind of canary in the coal mine in wild deer, signaling perhaps bigger health issues in those populations.
Yeah, it really highlights that complex dance between the bug's own resilience and invasion tricks, the environment, and then the critical role of, well, human management on farms or underlying health stressors out in the wild in deciding whether just being exposed actually leads to sickness.
And the learning definitely doesn't stop here. It sounds like while we know a lot, there are still important questions.
Yeah.
Researchers need to keep expanding surveillance, right?
Yeah.
And maybe dig deeper into the immunology, why some animals resist better than others.
Exactly. Comparative immunology would be fascinating. And using modern genomic tools to track specific strains, seeing how they move between wildlife, livestock, food, and people, that's crucial. Plus, faster, better diagnostic tests would always be helpful.
All aimed at improving how we protect animal health and, ultimately, reduce the risk to people.
Precisely. Enhancing our ability to diagnose, manage, and prevent listeriosis benefits everyone involved in that interconnected system.
So for you, our listener, maybe here's a final thought to chew on. How often do these animal diseases, things that might seem straightforward at first glance, actually reveal these really intricate connections? Connections back to our environment, how we manage things, even our own health? What other hidden stories might be lurking within seemingly simple diseases, telling us about the health of our animals, our planet, and ourselves?